Everybody can listen to me, okay? Okay, nice. So thank you all for coming. I'm very proud to be here and to have this nice audience. So I'm here to present my thesis. Please, can you turn the mobile phone on? Okay, so the title of my thesis work is Structure to Function Studies in Agri-cluster Proteins and Glucose 1-phosphate uridyl transferases. I hope in the end you know what these strange names mean. So let's start about the first protein I studied. That's the agri-cluster protein, HTP. So this is a protein with about 60 kilodaltons in molecular weight and has 3 domains. It has also 2 features. It's 2 iron-sulfur clusters, a cubane cluster, 4 iron, 4 sulfur cubane cluster, and a bit more complicated, we call it agri-cluster. This is the topology diagram of the protein, where you can see the 3 domains, the first domain in yellow, second in red, and third in blue. I want to mention that the cubane cluster is bound to the first 21st residues of the protein in this helix bundle over here in domain 1, while the agri cluster is almost in the geometrical center of the protein and is bound to cysteines from the 3 domains. Okay? So the topology diagram for the HTP that I just showed, and now I will give details about the agri cluster that is over here. So this is the agri cluster that is inside this protein. So it seems to be complicated, but there is a few features that I want you to memorize that will be important for the explanation further after. So the first is that there is this persisting residue that is a cysteine with an extra sulfur that is binding what we call ironate. And the second is this position over here, this X position. So let's describe the cluster. So the cluster is a square, and on the bottom, on the first layer, is a 2 iron, 2 sulfur layer. So it's Fe5, Fe6, S6, S5. So it's the first layer, and then it's a square, and then there has several layers on top. So the second layer is the oxygen here that we call X, and Fe8, and then on the third layer you have Fe7, O9, and O8. So these first clusters that were solved were for Vulgaris and were aerobically purified. And when this X position was not easy to assign an atom to this position because it was not sure if it was an oxygen, and also it was disordered. So the electron density was disordered and it could be modeled by 2 oxygen molecules over there. So after other studies were able to confirm that this was an oxygen by EXAFS and by resonance timing. So HTP distribution by organism. So HTP is distributed widely distributed in nature. It's distributed in eukaryotes and prokaryotes and archaea. You can see these 2 archaea. This is bacteria, and this is the structure we solved so far. Another interesting aspect is that many of the organisms that have HTP are organisms that are responsible for human diseases, like, for example, Clostridium tetani, the tetanus, or the well-known Bacillus anthracis. They have HTP in their gene. So the function is unknown. We don't know the function. But there are some clues. There is reports of hydroxamine reductase. So these people, Wolf and others in 2002. There is a related protein that is the carbon monoxide dehydrogenase that I'll speak a bit more later after. But they were able to replace the nickel that this protein has by an iron, and it got hydroxylamine reductase. So it got similar to the HTP. Also, the HTP transcription of the gene is regulated in response to nitric oxide. Oops. Fine. So we have the opportunity to see the new operating system of Windows, so it's Windows Vista. I'm not a really big fan of it, but well, it's the first time I'm going to see a boot of this operating system. Was fast. Yeah. Where is the mouse? Okay, so like I was saying, so the HTTP transcription is regulated by nitrate oxide. The HTTP transcription is related to a regulatory protein involved in the nitrogen stress response. There is increased anaerobic tolerance to hydroxylamine in E. coli cells when they are overexpressing HTP from Rhodobacter capsulatus, and wide array of organisms, many of which are pathogens that I've shown in the last slide. But at the same time, there was reports of peroxidase activity. The HTP gene is present in a large number of organisms that are not involved in the nitrogen cycle. HTP is induced by hydrogen peroxide, reports by Almeida and others. Induction to be regulated by redox-sensitive transcription activator involved in oxygen stress response. So this all brings us to these two clues. So HTP seems to be involved in protection against reactive nitrogen species and or oxygen-relative oxidative protection. So this is the two clues we have. So we have one known function, but we have these ideas. So these two clues that seem very different, they have something in common. The immune system in higher organisms, for example, has a way of avoiding pathogens that is using phagocytosis. So in phagocytosis, the immune system cells evolve the pathogen, and then they try to destroy the pathogen, or using digestive enzymes, or sometimes using a respiratory burst that frees free radicals. These free radicals, many of the times, are reactive nitrogen species or reactive oxygen species. So we don't know the function, but there is these clues that could bring potential interest in this protein. So what are the questions that we have to solve here? So the first is the structure that we determined for the ivory cluster, is it an artifact of oxygen because it was purified? The first structures that were published were purified in the presence of oxygen. Is it an artifact or is it real? Also, we want to know if there are structural changes upon reduction. When we reduce the protein, if there are changes in the cluster and in the protein. We want to know what are the substrates of this protein, and we want to know what is its function. So the first question was to see if it was an artifact of oxidation. So what we did was two independent studies, one done in the bacteria, in the HTP from bacteria, Succovirid sulfuricans, done mainly by Sofia Macedo, and another done in the Vulgaris, done by us. And we purified the protein in anaerobic conditions inside the glove box with the oxygen concentration below 5 parts per million, and we crystallized the protein also in the anaerobic atmosphere in nitrogen. So with this, the protein didn't see any oxygen from after expression, purification, and crystallization. So we knew that here, this cluster hadn't seen oxygen, could not be an artifact of oxidation. And this brought us, this was what we had in the oxygen when it was solved when it was purified in the presence of oxygen. And the structure for the sulfuricans and for Vulgaris is very similar to the structure for the cluster. And just to recapitulate, so we have the cysteine binding iron-8 and this oxygen position over here that we call X or oxygen, and two oxygens on top, 8 and 9. So, but this structure that was anaerobically purified has only one position for the oxygen, in this position over here. It's a bit spread out, but it's not two positions. And this happened in both the sulfuricans and Vulgaris. So when it was purified anaerobically, this position of this oxygen was unique. There was not disorder. So also, if we superimpose the Vulgaris and the sulfurican structures they are very, very similar, as you can see. In black, we have the structure of vulgaris, solved by me, and in gray, we have all the other structures of sulfuricans that were solved. Reduced and oxidized. They don't change very much overall. So, this is the anaerobic structure I just showed. Here, again, we did independent studies in vulgaris and in sulfuricans, and we published a nice paper together with Sophia. And I'm just going to speak about the vulgaris. That was the work that I did, but Sophia did the work in sulfuricans, and the results are almost the same. So she got the same conclusions. The only difference is that my structure was solved by multiple wavelength anomalous dispersion, and she solved their structures by ab initio. So this is the structure of an anaerobic purified vulgaris. And what we did was in the crystal, we soaked the crystal with excess of dithionite. So it was a very, like 30 millimolar of dithionite, very strong. The crystal resisted, and we were able to collect data from it. And this was the cluster of the reduced state structure. So if you look with me, you'll see the main difference. So the main difference is the persistine disappears, and this sulfur moves to the so-called X position. So here has a sulfur atom. Also, because of this disappearance of this sulfur, the iron 8 had to move to do a new covalent bond to the S gamma of this cysteine. So because of this, the iron 8 moves, and the glutamate goes after the iron 8. So, and these are the main changes here in the cluster of the irons. And then these two oxygens, O8 and O9, we have a unique oxygen species that is no longer at distances good for covalent bond, probably hydrogen bonded. But it's hard to say because the density here is not perfect for this Y position. So this is showing again the oxidized structure and the reduced structure in a simplified way. So you see the persistine that no longer disappears with the reduction. The sulfur that is here moves to the X position. You see it here. And the two oxygen O8 and O9, now it's only one oxygen species over here. The base changes a little bit. So then we have another data set that we had that was collected by Sophia, and it was a data set that was not possible to interpret the density near the hybrid cluster. People thought that it was a mixture, probably. So when people superimposed the oxidized structure on the electron density, they could not explain these large bulbs over here. But now that we know the anaerobic and the reduced structure, we could imagine that this A position was the movement of iron 8, that now exists a new iron 8 over here. We could imagine that this sulfur of the persistine moves to the C position, and this B position was a new position for the S gamma of this cysteine. So with this knowledge, it was possible to model and refine a mixture of reduced and oxidized structures. So I'm sorry for the quality of this image, but the electron density fitted perfectly. It's a bit complicated, but so we have a reduced and oxidized superimposed one on another, and the electron density fits perfectly. And also, if we do a difference map, you can see that the majority of these large bulbs of density that were not explained now are explained, with exception on the Y position. There is something there that we don't understand very well, and here this is visible. So we got the perfect fit, and we refined this with shellac, and we were able to refine 65% of the oxidized and 35% of the reduced. So then we have the COD comparison. COD is a carbon monoxide dehydrogenase. It's a protein that catalyzes the reaction of CO2 to CO. So this protein has sequence similarities with HTP, and the structural similarities also. So what the first thing we did was to superimpose the HTP that is in black to the CODs that were published in the literature. So what you can see is that the proteins superimpose very well, with the exception of this alpha helix that doesn't exist in the CODs, only exists in the HTPs. So this is the Polish diagram that I showed before. So this is the helix bundle that does exist in the CODs, and it has the cubane bound. So maybe the CODs don't have this cubane. Well, actually, some of the CODs dimerize, and when they dimerize, they get back this helix bundle from the monomer B. And then they have this cluster, this cubane cluster that is very similar to the cubic cluster in the HTP, and it's at the same distance from the aggregate cluster, probably transfers electrons to the aggregate cluster, as the in the HTP. Also, the CODs have another cubane, but it's completely different, that is absent in the HTPs, and have another cluster that I didn't put here for reasons of clarity. Still, this cubane, if they dimerize, it appears here. So this is the superposition and the cluster that the 3-unit bundle that disappears. So, if we look at the clusters, so this is the cluster of the anaerobic structure that I already explained. This is the cluster of the reduced structure, and this is the cluster that exists, the hybrid cluster that exists in the carbon monoxide dehydrogenases. So if you look carefully, the cluster has some similarities. So it has the same square base of 2 irons and 2 sulfurs as our structures. Here in this position, that is the position of the X, we call X or oxygen, that is a sulfur in the reduced and an oxygen on the anaerobic, has a sulfur atom. So in this particular aspect is more close to the reduced structure. Also, the iron 7 that exists over here does exist in the CODs and is bound. There is an extra iron bound to the base that does exist in the HTPs. But one interesting point is that, like I said, if they were able to replace this nickel by an iron, some people, and they got hydroxylamine activity when they replaced this nickel by an iron. That seems more similar to the iron 7. So if we superimpose these two structures, you can see that the base is quite fits very well. So I must say that I didn't fit the clusters actually. I fit the whole proteins, and then the clusters appeared very in the same positions. So you can see that the base is almost equal. This iron 8 is more similar to the anaerobic structure. This was the reason I used the anaerobic, because the iron 8 on the reduced moves a bit, and then there is this extra iron over here, and this is the nickel. So the ligands to the hybrid cluster in D. vulgaris, in disulfuricans, they are structural homologous to the ligands in the COD hybrid cluster. So if we look, for example, to cysteine 312 and to cysteine 355 in thermoacetica, cysteine 312 is that one that is binding iron 5, cysteine 355 is binding iron 1, so they are equivalent. And the same happens to all the ligands of the ions. So, if we, in light of these structures, if we were to think in a reaction, I will not say a mechanism because we don't have enough information for it. If the COD catalyzes the reaction like CO2 with a reduced protein receives 2 electrons and produces CO, if we imagine a species, for example, let's think that this species is H2O2, as some people have reported peroxidized activity. So if it's H2O2, this molecule will bind to the Y position, and then we'll receive 2 electrons from, this is red is reduced, we'll receive 2 electrons from the protein, we'll leave the 2 oxygens and get out as H2, H+, 2 atoms of H+, and then this structure could be reduced and get back to the reduced structure. So with this oxygen that was here maybe moved over here, the sulfur would move to the persistine, so this could explain a peroxidized activity here. So this is the anaerobic and the reduced structures. So conclusions. So we have shown that the anaerobic purification may rise to a different oxidation state where the oxygen does just have one position and is not disordered. And we have showed that this cluster is not an oxygen artifact because this protein didn't see oxygen during purification. We have shown that the reduced structure produces major changes in the hybrid cluster protein, and we can rationalize this change in a probable oxidized catalytic function. We have shown that probably this protein has a common ancestor with the CODs and probably a similar mechanism as the CODs. And future studies that should be done on this protein will be continuing to search for substrates because we still are not really sure if it's hydroxylamine activity, since the optimal pH is 9, and it's strange for a protein has an optimal pH of 9. But also the peroxidase activity has a good Km, but has a low Vmax. So could be that the reductor used to reduce the protein was not optimal. So we cannot be sure that these functions are the true functions physiologically. So we have to continue to search for substrates. And also, something that I didn't have the time to explain here, but it should be interesting to solve the structure of the HTP from M-thermo autotrophicus. This is a structure that lacks about 80 residues in the first domain of HTP, so maybe it also dimerizes as the CODs to produce the same fold. So and also this structure has an extra domain of rubidoxine, so has more information that it brings more input to the search for the function of this protein. So this is the end of this protein. And now I'll speak about UGPG. UGPG is glucose 1-phosphate uridyl transferase. This protein, we know the function. So this protein catalyzes the reaction of glucose 1-phosphate in uridyl 3-phosphate to UDP glucose. This is a reversible reaction, so it should be 2 arrows. So the protein can catalyze the reaction in both directions. So this protein that we studied is from the bacteria Pseudomonas elodea. That is a bacteria that was found because it produces GLAN in high quantities. I will explain a bit later what is GLAN, but it's a bacteria that is used industrially to produce GLAN. So they grow the bacteria, and the bacteria produce GLAN, they purify the GLAN, and they use the GLAN industrially. The pathway to the production of GLAN is partially elucidated by Sakurai and others. And catalyze the reaction between, so the reaction is glucose 1-phosphate to UDP glucose. This is our protein that we are studying. I should remark that there is a related protein that is RNLA that from glucose 1-phosphate and TTP can produce TTP glucose. So it's a very similar reaction. TTP and UTP are similar substrates. So this protein also has something like 30% sequence similarity with UGPG. This will be important later. So what is GLAN? GLAN, you know probably what is GLAN. It's E418. You don't know what's E418? You have to start seeing the things you eat in yogurts, etc. Probably you have E418 there. It's GLAN. It's an exopolysaccharide that is just to gellify, to make this gellify and stabilize. And it's approved in US and EU for pharmaceutical, food, and other uses. So it's in US, there is a big market for the production of GLAN, and they use this bacteria to produce it. So we did the expression of this protein in native and selenomet reduction. We also had to stabilize at high concentration because this protein, when we started to concentrate it, started to precipitate. So it was a bit hard to get it stable. We have to change solvents and find the proper conditions for it. And then the hardest was the crystallization. We got crystals very fast, in something like 3 to 6 months, but the crystals were not suitable for X-ray diffraction. So we had extensive use of robots, but when we were desperate, we started to do chemical methylation with borohydride to the lysins to put methyl groups, because there are some people that did this. This didn't work. And we also tried point mutations or arginines to alanines, and we were able to do some mutations, but we had to simulate the arginines that were at the surface simulate the structure, and then mutate one, and then from the result we had to mutate another and another, and this was a very hard work. And fortunately, in the middle we got a crystal that diffracted as it should. So we collected data on this native crystal, on the selenomet crystal, but also on a crystal soaked on potassium osmate. And from these 3 data sets, we were able to get the structure. So the structure is this one, is a tetramer. And from here, I mainly want to show you that we co-crystallized with glucose 1-phosphate. So this is the glucose, 4 glucoses because it's a tetramer, right? So this is a tetramer, but it's a tetramer that is composed of a dimer of dimers. Let me see if I can show you this. So if you look to this interface is much more packed. The protein, this is a view like from the bottom. You can see that the proteins interpenetrate, they enter one inside the other. And you see this V of helices, and in green and in magenta, they do a V like this, and they pack like this, and then they are hydrogen bonded. So this is quite strong. So this surface is strong. So what we think is that this is a dimer, and this is another dimer. So this is a dimer of dimers. While if you look at this interface over here, and this is the view from the right, you can see that the proteins just interact a few hydrogen bonds. So it's much less strong, the interaction. So, okay. So let's look at the monomer. So the monomer is shown here. This protein we have divided for reasons of clarity of the explanation in subdomains. We cannot really divide this in domains, but if we divide this in subdomains, we have clearly sugar binding subdomain, and we have glucose 1-phosphate bound there, so it was easy to identify. Then in green, we have the nucleotide binding subdomain, the domain where the NTP, in this case UTP, should bind. And we also divided this V, strange V of alpha helices in another subdomain. So if we look at the active site of the glucose and on the active site in general, you can see that the glucose is bound there. If we zoom the glucose, you can see the hydrogen bonds of that glucose makes to the protein. So you see here this hydrogen bond with D134, also the main chain of valine 204 and glycine 172, also the side chain of E191, and a water molecule. So because of this, we could assign here these residues. These residues, I will explain a bit later why they are here. But there is still space here for nucleotide. Also, the crystallization conditions were with 5 millimolar of G1P, so the G1P is there, but the crystallizations were in acid pH, pH 4.6. So at this pH, the side chain, the majority of the side chains of the protein are protonated, and as some of you may know, the binding of nucleotides is usually magnesium dependent. You need a magnesium site there. So if the side chains are protonated, magnesium is positive, cannot bind, and we think this is the reason our co-crystallizations with the nucleotides never worked, and we could not get crystallization conditions in high pH. So we think this is the reason. So let's see a comparison of UGPs with RMLA. So this is the reaction of UGP, the reaction of RMLA, as I explained you before. So UGP is very similar to RMLA. This catalyzes the reaction with G1P with UTP to give UTP glucose, and the other with TTP to give TDP glucose. So we're comparing with this protein over here. So if we superimpose the monomers, you can see that they are very, very superimposable. There are two main differences that we can see immediately. The first one is this V alpha helices that doesn't exist in RMLAs. So it's the helices, the first helix exists, but then has a 3 helix bundle over here that is a well-known allosteric binding site for the nucleotide in RMLAs. That it seems that UGP doesn't have it. The second thing you can see is that all the RMLAs have this loop closed, and the active site is over here. I don't have here the glucose. So it seems this loop is a gate and is closing on all the RMLAs, but our loop is open. So this is the two main differences. The rest of the protein is quite similar. So we don't look to the table, but just to show you all the RMLAs that are here is from Salmonella enterica, Escherichia coli, Pseudomonas aeruginosa, and M. thermophilus. So, and there are structures with different substrates in. So the difference, if we look to the tetramer, I don't have here a picture of the tetramer of the RMLA, but I have a scheme, and the tetramer of the RMLA is different from the tetramer of UGPG. So that it's like there is a swap between like these two, the cyan and yellow swaps with the green and with the magenta and green. And this swap, what does it mean? It means that the dimer still, the dimer is the same. So the dimer here, we call it dimer on these two monomers. For example, the magenta and the green still exists. So the dimer is the same. So we still have a dimer of dimers. But the interface inside the tetramer, that here was this one, here it's outside, it's to the outside of the protein and to the outside of the protein. And the opposite also happens. The surface of the monomers that are to the outside of the protein here, now they are to the inside. So the tetramer is built differently. So looking to the active site, so this active site I already showed, what we did was so remember that we have superimposed the structures. So if we grab one of the RMLA structures that had deoxythymine inside, the deoxythymine would fit on this empty space, and this is the deoxythymine in RMLA of one published by Zuccotti. So we were able to fit also the deoxythymine by hand on this empty space using this as a model, using this structure as a model, and we could see that our side chains were almost already ready to receive this molecule there. So we could see the same hydrogen bonding pattern that existed in the published structure. So this is a model, but it seems that we have space and we can identify the residues that bind. Also, for the residues that bind by the side chain, they didn't change, D, D, but the residues, for example, that bind by the main chain is the glycine, alanine, or here is the glycine. But if it's the side chain that binds the oxytinin, then the residue is the same. So that is homology. So conclusions. So we have shown that there is a different tetrameric arrangement between UGPGs and RMLAs. We have shown that there is an important loop gating the active site that is open in UGPG and is closed in all the RMLAs. We have shown that the active site comparison the active site is very similar and suggests a similar mechanism again with the RMLAs. Sorry, it should be RMLA. And we have shown that the UGPG doesn't have this secondary allosteric site that was reported in the RMLAs. And for future studies, we should find a crystallization condition at higher pH that we could have magnesium there and eventually could be co-crystallized with the nucleotides. And it could be interesting to do mutation studies in this important loop and see if the protein loses or increases its activity to see if this loop is really important as a gatekeeper of this active site. So this is all. And I would like to acknowledge all these persons, especially my supervisors, Dr. Carlos Corzão and Dr. Edward Mitchell. Also Peter Lindley, that supervised me while he was still in the SRF and after in ITDB. Maria Armeni, the head of the group, Sean Maxini. Also all the people in technical, where we collaborated in the glucose 1-phosphate uridyl transferase, especially Arsene and Rita, and all the rest, and also purification I did in the US. So also I would like to thank you all for listening.
Não está